We start this new second part of the first session with Alexandra Weil. It's a very pleasure to introduce Alexandra. Alexandra is a science leader in nucleic acid research group at LGC in the UK, and LGC is designed as a National Measurement Institute for Chemical and Bioanalytical Measurement in the UK. And you arrived in LGC at 2009. You developed digital PCR for a very, for a lot of application. So today, Alexandra talk about the digital to characterize viral vector genomes. Thank you, Alexandra.
Morning, everyone. Thank you very much for the introduction, and it's great to be here. And I'm going to be talking about viral vector genomes, so a very different kind of microgenomics area, but where we're looking at the individual genome within those viral vectors and how they're used. So just a very quick overview. I'm actually going to explain what the National Measurement Lab where I work is, and really just so you can understand why we do the kind of research we do, because it's very different from what you might do in academia or in other industry labs or in hospitals. And I'm going to very quickly just go through how digital PCR works, because while it may be a term that many of you know, just to make sure that we're all at the same point. So when I show you some data, you can understand what you're looking at with the various graphs, and then just where digital PCR can support, because there's a large number of spaces within the workflow of viral vector production, but I'm going to focus very much on looking at the quality of the genome packaging within the viral vector that would then deliver the gene therapy product. So this is my lab. We're the molecular biology lab. We have organic chemistry, inorganic chemistry, cell biology, all within the National Measurement Laboratory. We are the designated institute in the United Kingdom for chemical and bioanalytical measurements. And essentially what that means is we get money from the UK government to support UK science, but we also work with the metrology community, that is the measurement institutes around the world, to support each other to understand measurements and metrology. So metrology is science of measurement. It's not a nasal gazing activity. It's where we actually drill down and look really closely at the sources of error in your measurement. So what is the truth? And then how are we measuring it? Because everything we do is essentially an estimation. How close is our estimate to the true value? And it's also an idea of traceability. So traceability is a slightly challenging concept in biology. It's very straightforward in physics and pretty straightforward in chemistry, but it's where we can follow the traceability of your measurement. And ultimately, and the easiest way to really explain it, is if you go to a hospital in, say, Göteborg, or you go to a hospital in Florence and you have the same disease and you give them a sample, they will both diagnose you with the same disease and you would get the same treatment. And so the traceability of your method allows that to happen, particularly across the international borders, rather than just within your institute. And so the ultimate game is to improve measurement accuracy. So this is a traceability chain, which can look very frightening and complicated, but ultimately up at the top, we have an SI unit, and down at the bottom, we have the actual measurement that matters. That is, in this particular case, a qPCR test in a clinical laboratory in a hospital. And so what we do with biology and as a metrology institute is we try and work out how we can do the traceability up to an SI unit. Now, in biology, that's very challenging. I've got the units up there, but essentially the closest ones we can get to are the mole or even the weight, so the kilogram, which are largely unhelpful when we're looking at nucleic acids, because it's the sequence that really matters. Someone with a large tumor is not more sick than someone with a small tumor. And so what we do is we develop methods and reference materials to support those methods. And digital PCR is one of the methods that we are using that we think you can use in a reference laboratory, because of the way it quantifies based on sequence that is not restricted to using a calibration to convert the instrument output into something tangible, like the copies or the percentage of the mutation. And so this is our goal within the National Measurement Laboratory in the UK, is to improve nucleic acid analysis. So just a quick overview on viral vector and gene therapy. Essentially, we're trying to treat genetic diseases by restoring defective functions. And we do that by either adding in the correct protein, the correct genetic material to make the correct protein, or we try and remove and replace a defective protein. So I've just got an example here of the CAR T cell therapy, but the other areas in the biomanufacturing area where you might modify a whole range of cells to do something useful for us. And so I've got the vaccine production, which is one of the big spaces at the moment. And so we use viral vectors, which are essentially modified viruses. We're completely taking advantage of the fact that viruses have evolved to infect cells. And so hopefully that gets the thing we want into the cell that we want nice and efficiently. And really, viral vectors are just encoding a gene within their genome that they then deliver for us. And there's a number of safety features, which I don't have time to go into, but ultimately, we remove like their ability to replicate independently. And so we've got a number of listed here. Some are retroviruses, but today I'm actually going to focus on some of the work we're doing with the AAV. So just very quickly, this is a wild type AAV. So this has everything it needs to get into the cell to replicate and do its thing. Where that differs from the viral vector is we have 3 plasmids that we've split all the genes onto, and the transgene plasmid is where between the ITRs, we put the thing we want in that genome. So this would be our gene of interest. And for the sake of this lecture, I just have GFP in my transgene at the moment. And then there are separate plasmids that contain all the replication machinery, the ability to make the capsids to surround those viral particles. And then we use packaging cells, and then we have to extract out those the recombinant AAV particles before they're then screened and checked and put into the patient. And there are now a number of critical quality attributes. So with a lot of the work, it's very challenging to actually work out the purity of these particles, how many of them are what we want them to be, how many are maybe empty, how many encode the wrong thing, because when we're looking at treatment, what we don't want to do is any more harm to the patient. So I'm now going to explain just a bit about digital PCR and then tell you where I think it's going to help in the space for the AAV. So it's a lot like real-time PCR. You would prepare, say, a 20 microliter reaction, and then that contains all of the components that you need to do your real-time PCR. And then instead of cycling, you do this extra step where you split that sample by dilution. And this is called a limiting dilution. And so in this schematic here, I've got, rather than one 20 microliter reaction, I've got 21 microliter reactions. And what is absolutely crucial here is that some of those 1 microliter reactions do not contain a nucleic acid molecule, which is the little black lines that I've got in the tubes. Then you do your PCR cycling. Oh, sorry, clicked on too fast. And then you get fluorescence wherever you had an original molecule. So we're no longer interested in the exponential increase, we're just interested in that endpoint. And what we do is we then look at the particular reaction and we count up how many are positive. And digital PCR gives you a quantification value based on the proportion of those positives. So how can it support the viral vector characterization? Well, it's literally you take that proportion, you apply Poisson statistics, which accounts for any partition that maybe contained more than 1 molecule at the beginning, but ultimately that is your output from your instrument. So we don't have to have a calibrator that says, right, this level of fluorescence means this amount of starting material, which means we don't have that huge challenge of what do we use as a calibrant. We can just put in our sample and measure it. Because we can follow this statistical distribution, the Poisson distribution, it's a non-normal distribution, but it has very specific precision to it. We can look at the standard deviations, we can look at the 95% confidence intervals, and it produces it's very reproducible measurements, which I've just shown here. So these are the 3 digital platforms that we have in our lab, and this is, I'm running the same units of material here with the same assay, and ultimately, what we're interested in is having a nice big gap. So these are the positives up here, all 3 of these instruments colored them blue, and these are the negatives down here. And each of these tiny little dots is a partition. So all of these instruments here now use microfluidics to take your 20, 40 microliter reaction and subdivide it into sub-nanoliter reactions. So we're looking at kind of the sort of 10 to 20,000 partitions that we can see here. So we've got lots up here and lots here, and I've got some NTCs in here as well. And so we get this really lovely separation. But what is totally awesome about this is when you do all of these separately, we only get about a 6% difference between the platforms. And while this was in our lab, we actually had different operators generating this material. You find that when you use a method like real-time PCR, you could actually be looking at log differences between different laboratories because they have different instruments, etc. And all of those put in huge amount of error because you're looking at the exponential increase in amplicon and you need some calibration. And what we have here, beautifully, is showing that even between your platforms, you get really good reproducibility. So the SI traceability of 1 is really just trying to trace it back up. The measurement, I showed you right at the beginning, the traceability chain, up to show how it's traceable to the SI units. This is a huge area that we're looking at in the international metrology community, the international metrology community. So how can this actually work? Well, with your viral vectors, they're very bespoke, yeah? So if you produce one batch for one particular vaccine, your transgene between your ITRs is completely different to another batch, which is maybe being generated to do a totally different function. And so the idea of how we can use sort of quality control and design assays is pretty challenging. But what we have, if you remember the image that I showed you, is we have that P transfer vector, which is a plasmid. It's DNA, it's double-stranded. The AAV genome is single-stranded, but this is an excellent surrogate for the genome in order to work up our assays and check that everything is working beautifully. And so really what we actually want to do is find those viral particles that have the correct genome, they have the genes in the right order, it's packaged correctly, and so we can start to get some of those critical quality attributes with quantitative data. So we can start to say, this particular batch is 40% correct in identity, and then you can start to modify your process or work out how you can remove the 60% that aren't correct. And what we're going to use here with digital PCR is the linkage of the targets. And I'm actually going to run through some slides that show you how this works. So just before I do that, this is just the general workflow. And we're going to talk mostly about this today, the quantity and the quality of the viral titer. But what I really like about the method that we've been developing is there's actually multiple points that the same method can be used to validate. So we can actually look right at the beginning. I apologize, this is falling slightly off the bottom. But when we can start to look at the actual transfection efficiency into those helper cells, if we don't get all 3 of those plasmids in, we won't make as many AAV particles. So it's part of that biomanufacturing. How can we improve that process? Big area that we are just starting to look in is the kind of carryover, the cell contamination. So the AAV particles are put out into the supernatant where you collect them and you clean them up and you purify them. But maybe there's some leftover host DNA or any kind of debris left over from those packaging cells, which may cause harm to the host when you put the particles, the AAV particles, into the patient or the host. Then we can actually look at the delivery into the cell. Again, we can still target the AAV genome. And then in the case of lentiviruses that we've been doing for the last few years, you can actually start to look at the integration efficiency into the host genome. But I'm going to focus today on that very first one that I showed you, which is the viral titer and the quality. So at the National Measurement Lab, whenever we're presented with a new kind of analyte, we sort of step back and we have a look, right, what do we want to measure? We want to measure the viral particles. But actually, we can't really start at that point because it's complex. We've got capsids we need to get rid of. Also, where do we get the AAV particles from that are 100% identity in what you want and 50%? There's no reference material. So there's nothing really we can use that we know the truth of to check that our method's working. So what we do is we go simple. And so what I've got is as we go right, we're going through the various guises of that genome where the sequence is identical. So we've got our original vector that goes into our producer cell. We've got the single-stranded genome that is then packaged into that capsid. And so we started with the P-transfer because everything between those two ITRs ends up in our viral particle. So the method development. We took the P-transfer, this one encodes an EGFP, and we cut it so that it was linearized, so it sort of resembled the AAV genome. Now it's double-stranded. We looked at various things of trying to denature it, trying to get it single-stranded, but then we had sense-strand and sense-strand, and it got a bit complicated. So we decided to stay just with the linear plasmid. Now, of course, we've lost the secondary structure that you would have with, oh, sorry, I'm clicking forward too fast. Lost the secondary structure that you have with the ITR, but let's start simple. Let's get that ITR assay up and running. And so what we can do is start to look at the ratios. Now we're using the plasmid, we're expecting everything to be 1-to-1. But we've got something interesting with the ITRs. We've got two ITRs, but they're on the same molecule. That is, they are linked. And so what happens is when you do that digital PCR, the whole molecule goes into one partition. So I've just got a little schematic here where each of these circles is just a partition in the digital PCR. And what happens then is if we use a separate assay to target each of those three targets, so the ITR, the poly-A assay, and the EGFP, we get the signal come out, but for the ITRs, we will underquant. We will only measure half of those ITRs because we can't actually see the two different ones. This is just an example of the output where we would do some assay optimization. You can see that the three assays are working beautifully. You can see that the peak resolution, that is the distance between the grays, so the negative partitions and the positives, is different. That is actually okay. That is one of the nice robust things about digital PCR, is you can quantify the poly-A and the EGFP despite there being a slight difference. You can overcome that, you can add a bit more probe, but actually this is completely fit for purpose. And this is what we get for the copy number concentration. So taking those positives and then using the dilution factors that you use to get into the reaction, we can work out the copy number. And we put in roughly 40,000 plasmid copies, and you can see, hopefully from this graph, that the EGFP and the poly-A copy number concentration are pretty close to each other. They're close to 1. And what I've got here is these are the average of an experiment, and then we've got about 9 different experiments with different people in our lab. So we've got a huge amount of technical variability in those reactions, and yet we are measuring pretty close to what we are expecting, which is 40,000. Now, what was a bit weird was the ITRs was lower. We were expecting it to be higher. We thought maybe some of our molecules got fragmented during the partitioning, but actually it's a little bit lower. And we were sort of wondering, it can't be secondary structure because it's plasmid. So what we thought was, well, we'll just go ahead and let's separate those two targets. So we did another restriction digest. With digital PCR, you can actually add the restriction enzymes into the reaction, and then it just happens in the sort of 15 minutes that it takes you to sort of set up before you prepare your partitioning. And so actually it ends up partitioning like this. So when you do the ITRs, you get double the signal. And so if we just have a quick look at these graphs, and they're the original 3 from before. But what I want to show you from here is the enzyme hasn't disrupted. I've checked, it's not causing inhibition, it's not changed the profile of where the negatives sit or the positives sit. And then when we look at the copy number concentration, it was so perfect, it was awesome. I mean, it doubled up to about 80,000. And it didn't really matter which restriction enzyme we used, so that was great because that gives us a little bit more flexibility in the future. So at this point, we thought, super, right, we've got something working in the plasmid, not hugely exciting, so let's go and actually have a look at some of our AAVs. So we started with some genome extracts. So remember, we don't really know what we've got here. We have made those AAV particles, then we've purified them and extracted out the single-stranded genomic DNA from them. And so what we did was just with one type, we did 4 flasks. So we started looking, let's start with the biology. This is what we do at the National Measurement Lab. Let's chuck in as much biological variation as we can find. So we did 4 flasks, and we extracted them. So this is an AAV2 model. There are many serotypes of AAV. We used a fairly standard kit, and then we quantified with digital PCR. In this case, we changed the restriction enzyme. ECOR1 and HinD3 don't cut single-stranded DNA, so we changed to MSP1, which will cut single-stranded and double-stranded. So we thought this would be a good method. And we're remembering that our ITRs have now got that secondary structure at the end. And these are our results. And what's really interesting about this is, firstly, the biology, we've got different results for the 4 different flasks. Now, of course, you can introduce the technical variability. They're physically 4 different flasks, so maybe flask number 4 is out of the incubator for a bit longer, it takes a bit longer to process, and this is what biology does, right? It's a little bit compounding. But ultimately, what we found was, between calculating the gene ratios, we always had less EGFP than poly A. The ITR copy number was always higher, suggesting that we've got viral particles with ITRs in them, but not with your gene of interest. And what we found was the MSP1 did increase the copy number. Now, it didn't double it. The numbers that I've got up on here is the ratio between the ITR without digestion and the ITR with digestion. I've also got the P transfer here. That's starred because it's on a totally different scale. It's 40,000 and then up to not quite 80,000 in this context. We think it's because MSP1 doesn't digest double-stranded quite as well as something like ECOR1 and HinD3. But ultimately, this started telling us, oh, actually, the mass spec methods that they're using to look at the empty-full capsid ratios in AAVs, they are, these are good methods. They are suggesting that, yes, indeed, you do get AAV particles, which are only partially packaged. So the next step we did was to develop a multiplex method. So that was all with uniplex, so we've got quite a lot of variability. They're all in separate reactions. And we switched instruments at this point. So we moved from the QX200 and we moved to the QIAcuity. We also designed a couple more assays. We might as well use all the colors that we've got. So I popped in a CMV here, and we've also got the WPRE, and we put those on different colors to be detected in different channels. We verified they all worked in uniplex. We checked that it transferred beautifully from the QX200 to the QIAcuity, which of course it did. We knew it would. And this is the kind of output we got. So this is similar to what I had before, but it's a 2-color plot. So we've got, in this particular example, I've just got 4 of the assays showing. I've got one assay, the fluorescence, along the x-axis, and one up the y-axis. I've kept in the fluorophores, and I've kept it kind of colored based on the channel, just to sort of help it be a little bit more visual. But what we get is now a quadrant. So we get 4 possibilities in each of the outputs in each of the 2D plots. So we've got, these are the empty, so they're empty for both. These ones are positive for just WPRE, these ones are positive for just ITR, and these ones are positive for both WPRE and ITR. And it's the same up here. And what you can see is the separation. This was our first shot at it. We tweaked it a little bit more. We've actually increased that separation because I wasn't hugely happy with where that line possibly dissects through some of the negatives. But we started looking at this, and what we also found was we started getting sort of extra clusters, and we looked into this, and what we think those extra clusters are is, in this particular case, this is where these ones here are positive for the ITR and the WPRE, but we also think they're positive for the EGFP, and we can't see that in the 2D. We'd need to go 3D or 4D or somehow, and you can spend quite a lot of time going down a rabbit hole trying to work out and spatially work out what's going on. But ultimately, it gives us a huge amount of information from single reactions. So I should say at this point, we didn't want to restrict ourselves to just AAV2. So we started chucking in, oh, it's slightly going off the bottom, but we started putting in different, oh, time's up. No, I've only got 2 more slides. Anyway, we started developing other assays, so other poly-A's, so we had SV40, we did some other promoters, really just so that we could start to look at AAVs that are going to be working with under different conditions in maybe different host cells. And the idea was to try and get a plug-and-play approach where you could sort of go, right, I'll take that assay, put them in the reaction. And ultimately, the fascinating thing about digital PCR, and because you have these partitionings, that sounds like a totally crazy thing to do, and how you go about optimizing it sounds completely bonkers. But because you have that partitioning, you... that's great, thank you. I'm on this slide, so that's great. So because you have that partitioning, it really does help you do the multiplexing. It makes it a lot more straightforward. So what we wanted to do was then exploit the linkage in those genes, because we're using a 5-plex. Any partition that gets the genome in there, which has all of the targets, so that is an understanding that we've got a fully packaged genome, it should be positive for all 5 targets. So we're going to look at it in all the channels. We can also start putting numbers on it, like the percentage of the linkage, so the percentage of the molecules. And there's a very nice paper back in 2015, Regan et al. With the PLOS1, where they've actually said you can start looking at how you get a disproportionate number of double positive partitions compared to the singles. And so here's my little schematic with how this might go. So we've got a correctly packaged, this is what we want, this is not what we want. We think there are a number in there which have ITRs and then are empties. And so we get something that looks a little bit like this. That was obviously did have a molecule in it, but it's got lost somewhere. And we get results that look like this. So what I've got here is I'm just showing it with the plasmid at the top, because we know that the plasmid should be 100% linkage. And what I've done along the x-axis, it's fallen off slightly, but that is the distance between the two. So I've just popped the schematic up at the top so you can see that there's varying distances between them. And ultimately what we get is a really nice curve where it sort of plateaus off around about the 80% of having those full-length molecules. Now what I've got here in just green and orange is the first 2 of those single-stranded DNA extracts that we use for the AAVs and how they responded. And you can see that it's much lower, and it kind of correlates at the moment. This data we got last week, so I just chucked this in at the last minute. But what I want to sort of almost explain to you as well is it came off the instrument last week, we've had a look at it, and we've managed to work out roughly what the percentage of the linkage is. So I just want to finish with the really exciting work that we have also just started doing, where you just add the viral particles into each of your partitions. And so you're looking at it in this way, 1 viral particle per partition. So it's just presence, absence. We've got rid of that complication with the ITR and like whether it's a double copies. But we're going low concentration at the moment. So we're going very low, because what we don't want to risk is having 2 AAV particles in there. And that's certainly something that we're doing in the future to try and work out if we can expand the dynamic range of what we've got. And we just burst them in that initial 95 degree. I said we didn't need the MSP1, and we get something like this. And this is where we started looking at different AAV sources. So we chucked in 8 and 9. Sorry, that's not a very scientific term. We selected AAV8 and AAV9, and we had a quick look. And this is what we got. And this is the first time that we actually started being able to characterize our different AAVs. And what you can see is actually there's a large number which just have the ITR only. The parts in red is partial, so we've kind of got the ITRs and something else, but not quite everything. But what we've got is quite a large number of the full. And so at this point, I apologize for going over. I'll just finish with my summary of what I've shown you. So digital PCR, it's a powerful tool. We use it in lots of areas, not just viral vector analysis, but it very much helps us with that bespoke nature of the viral vectors, because we can develop the method, we have the materials to develop the method. We've got this new platform, so we can do the greater multiplexing, and it's reasonably uncomplicated, and it was pretty straightforward to validate it. So the method development was with the P-transfer, and it directly transferred into measuring the single-stranded genomic DNA and the viral particles. And that initial look at the plug-and-play strategy does seem to be a nice, flexible way of working this through. So just finishing with the future work, we are expanding in this area. We are looking at that digital PCR as the reference measurement procedure. One of the big areas I look at is actually using it to support method development. I'm very interested, you transfect your cells with like 250 nanograms of DNA. Well, what does that mean in terms of copy numbers? Can we refine it? Can we improve that transfection so we don't waste material, we don't waste cells? but also the purity, I said. So when you get those AAV particles with the how much of your production cell is running over. And so the last slide is just my acknowledgements. There we go. And I'd just like to acknowledge Theo, who is the PhD student at the University of Kent, who's done much of this work with me, and the rest of my colleagues.
Thank you, Alexandra. Great talk. Some questions?
Thank you. I have a question. So as well for this investigation, one uses analytical ultracentrifugation. Did you try to correlate the analytical ultracentrifugation data with this? And the second question, how do you calculate the proportion of the empty particles if they appear?
So I'll answer the second question first. The empty particles are blind to us. We can't tell the difference between a partition that contains an empty particle and a partition that contains nothing. So that is one of our limitations, absolutely. The first one with the ultracentrifugation, we have started looking at the method development for the purification of those particles. It's a really key area. And some of the AAV samples that we have gone through that ultracentrifugation. And we will be very interested to look at any kind of carryover from host cells as well as the actual purity. Do they sit differently in the gradient depending on how heavy they are? So yeah, no, that's it's definitely what we want to use our method to help validate that kind of process.
Okay.
Thank you, Alexandra, for the talk. Concerning the direct measurement of viral particles in the system, I would like to know what kind of system you used for that, and correlate question regarding the possibility to make measurement on single cells isolated in nanowells or emulsions. And do you think it could be possible to also measure viral particles inside the single cells, if it's possible?
That's what I want to do. So first question, oh, sorry, I got overexcited by your last question. I'm sorry, I've forgotten. The first question was, we're using the QIAcuity. So the first question was what instrument? We use the QIAcuity. So it's a plate where we can just set up the plates in a biohazardous hood, and we can just seal it, and then the instrument takes care of all the partitioning, cycling, and reading. We've just got single cell technology, and actually it's a really exciting area that we can start to look at. I mean, I'm interested in looking at bacteria. How many plasmids do you contain? Is the number maintained? Like, how does it vary? So when you're looking at sort of gene circuits, how are you changing the gene circuits in error just because you've changed how many plasmids? And one of the other big areas, of course, is looking at the mammalian cells with the different viral vectors in. Like, can we actually get a measurement using digital PCR and single cell isolation techniques to literally count how many particles are being produced in those producer cells? And then can we tweak it? Can we make it more efficient? Because we want it to be as high as possible, keeping our cells happy, working well. But it's time, money, and resources. So in the biomanufacturing, it's a crucial area of that workflow.
Thank you very much. Sorry, we don't have time for other questions. Thank you, Alexandra.